Welcome back, everyone. So we're going to get into Chapter 7 in our OpenStax textbook and study patterns of inheritance this week. If you are in Campbell Biology, this will be chapters 11 and 12, starting with chapter 11 in this video. So in this unit, we're going to study genetics. So genetics is the study of heredity. So we're going to start out talking a little bit about Gregor Mendel. So he's basically known as the father of genetics. So, he, in the 1800s, before we even knew what chromosomes or genes were, he started to kind of put together this understanding of inheritance patterns, dealing with just the simple pea plant. So thinking about how, we can get violet colored flowers versus white colored flowers through simple inheritance patterns that he revealed through some experimentation with these pea plants. And so based on his experimentation, he was able to figure out that the pea plants actually carry two sets of genetics, and based on that information, he came up with these ideas, which set up this understanding of genetics that we know today. So we're going to start out by looking at 7.1 Mendel's experiments in this video, and then we'll build on that in subsequent videos this week. Okay. So by the end of this video, you should be able to explain the scientific reasons for the success of Mendel's experimental work, as well as describe the expected outcomes of what's called a monohybrid cross, which involves dominant and recessive alleles. So we're going to start out with a crash course video. So this crash course video will give us kind of an overview of heredity. So he'll go into the history of our understanding of genetics with, Gregor Mendel and his work, as well as basically some of the terminology that we're going to be using this week, as well as, some of the stuff that we're going to learn in chapter 11 and 12 this week. So, he'll go over some of the information that is covered in 7.1 as well as 7.2 and 3. So, we're getting ahead of ourselves, but this will be a good kind of overview of the topic this week. Okay? So let's get into that video.
So I have this brother, John. You may have heard of him.
Hi there.
And as it happens, John and I have the exact same parents.
Yes, Mom and Dad Green.
And since we have the same parents, it's to be expected that John and I would have similar physical characteristics because the source of our DNA is exactly the same.
Hank and I share some genes, but no one knew anything about chromosomes or DNA until like the middle of the 20th century, and people have been noticing that brothers tend to look alike since people started noticing stuff or whatever.
It was very scientific, John.
I will remind you that I am doing you a favor.
Heredity. It's basically just the passing on of genetic traits from parents to offspring. And like John said, the study of heredity is ancient, although the first ideas about how the goods were passed on from parents to kids were really wrong. For instance, the concept that people were working with for nearly 2,000 years came from Aristotle, who suggested that we are each a mixture of our parents' traits, with the father kind of supplying the life force to the new human, and the mother supplying the building blocks to put it all together. Aristotle also thought that semen was like highly purified menstrual blood, which is why we still refer to bloodlines when we're talking about heredity. Anyway, since nobody had a better idea, and since nobody really wanted to tangle with Aristotle, for hundreds of years everybody just assumed that our parents' traits just sort of blended together in us, like if a black squirrel and a white squirrel fell in love and decided to start a family together, their offspring would be gray. The first person to really start studying and thinking about heredity in a modern way was this Austrian monk named Gregor Mendel. And Mendel demonstrated that inheritance followed particular patterns. In the mid-1800s, Mendel spent sort of an unhealthy amount of time grubbing around in his garden with a bunch of pea plants. And through a series of experiments crossing the pea plants and seeing which traits got passed on and which didn't, he came up with a framework for understanding how traits actually get passed from one generation to another. So to talk about classical genetics, which includes Mendel's ideas about how traits get passed along from parents to children, kinda have to simplify the crap out of genetics. I hope you don't mind. So we've all got chromosomes, which are the form that our DNA takes in order to get passed on from parent to child. Human cells have 23 pairs of chromosomes. Now, a gene is a section of DNA in a specific location on a chromosome that contains information that determines a trait. Of course, the vast majority of the time, a physical trait is a reflection of a bunch of different genes working together, which makes this all very confusing. When this happens, it's called a polygenic trait. Polygenic. Many genes. And then again, sometimes a single gene can influence how multiple traits are going to be expressed, and these genes are called pleiotropic. However, some, very few, but some single traits are decided by a single gene, like the color of pea flowers, for example, which is what Mendel studied when he discovered all of this stuff. And when that happens, in Mendel's honor, we call that a Mendelian trait. There are a couple examples of Mendelian traits in humans, one of them being the relative wetness or dryness of your earwax. So there is just one gene that determines the consistency of your earwax, and that gene is located at the very same spot on each person's chromosome, right here, chromosome 16. However, there's one version of this gene, or allele, that says the wax is going to be wet, and there's another allele that says the wax is going to be dry. You may be asking yourself what the difference is between these two things, and I'm glad you asked, because we actually know the answer to that question. Among the many amino acids that make up this particular gene sequence, there is one exact slot where they are different. If the amino acid is glycine in that slot, you're going to have wet earwax, but if it's arginine, it's dry. Now comes the question of how you get what you get from your parents. In most animals, basically any cell in the body that isn't a sperm or an egg, these are called somatic cells, are diploid, meaning that there are two sets of chromosomes, one inherited from each of your parents. So you get one earwax determining allele from your mom and one from your dad. And I should mention that the reason for this is that gametes, or sex cells, senior sperm and madame egg, are haploid cells, meaning that they only have one set of chromosomes. Again, for emphasis, non-sex cells are called somatic cells and they are diploid, sex cells are called gametes and they are haploid. This makes a lot of sense because a sperm or an egg has a very specific motivation. They're seriously hoping to score, and if they do, they plan to join with a complementary haploid cell that has the other pair of chromosomes they're going to need to make a new human or buffalo or squid or whatever. Also, just so you know, some plants have polyploid cells, which means that they have more than two sets of chromosomes in each cell, which isn't better or anything, it's just how they do. But anyway, the point of this all is that we inherit one version of the earwax gene from each of our parents. So, back to earwax. So let's just say that your mom gives you a wet earwax allele and your dad gives you a dry earwax allele. Good lord, your dad has horribly ugly ears. Anyway, since your parents have two alleles, each for one gene inherited from each of their parents, the one passed along to you is entirely random. So a lot of what Mendel discovered is that when there are two alleles that decide the outcome of a specific trait, one of these alleles could be dominant and the other one recessive. Dominance is the relationship between alleles in which one allele masks or totally suppresses the expression of another allele. So back to earwax, because I know we all love talking about it so much. It turns out that mom's wet earwax allele is dominant, which is why she gets a big W, and dad's dry earwax allele is recessive, which is why he has to be a little W.
Go Mom!
Oh, you're back.
Yeah, you sound surprised.
Anyway, mom's allele is dominant, and that settles it, right? We're gonna have wet earwax.
Something about the way that you said that tells me it's not that easy.
You are so much smarter than you look. It is indeed not that easy. So just because an allele is recessive doesn't mean that it's less common in all of your genetic material than the dominant allele, which leads us to the assumption, the correct assumption, that there's something else going on here.
I'm definitely getting that vibe from you.
So it has to do with mom and dad's parents, because everybody inherits two alleles from their parents. Mom got one from Nanny and one from Pawpaw, and let's just say that mom got a little W from Nanny and a big W allele from Pawpaw. That means that mom's genotype, or genetic makeup, when it comes to that single trait, is heterozygous, which means that she inherited two different versions of the same gene from each of her parents. Dad, on the other hand, is a homozygote.
Let me guess, that means you had two of the same allele, either a little W or a big W, inherited from both grandma and grandpa.
Right, and in order for this to all work out the way that I want it to, let's just say that both grandma and grandpa would have passed little W's down to dad, making his genotype homozygous recessive for this gene.
Okay, so I'm keeping score in my head right now, and according to my calculations, mom is a big W little w, and dad is a little w little w.
And now we're going to try to figure out what our earwax phenotype is. And phenotype is what's expressed physically, or in this case, what you'd see if you looked into our ears.
Oh, so we're going to do like a Punnett square or anything? This is why I do history. If we're doing Punnett squares, I'm leaving.
But I was just going to start to talk about people again. So Reginald C. Punnett, who was a total Gregor Mendel fanboy, invented the Punnett square as a way to diagram the outcome of a particular crossbreeding experiment. And a really simple one looks like this. So let's put mom on the side here and give her a big W and a little w, and let's put dad at the top, and he gets 2 little w's. So if you fill this in, looks like there's a 50-50 chance that any child of this mating will be homozygous or heterozygous. And as for our phenotype, shakes out the same way. John and I both have a 50% chance of having wet earwax, a 50% chance of having dry earwax. So I just had to go and call John, because now he's not participating, because he doesn't like Punnett squares. And it turns out that he has wet earwax. I also have wet earwax, which, is not that unlikely considering that our parents were homozygous and heterozygous. This may explain the odor of our bathroom when we were growing up, because it turns out that there's a correlation between wet earwax and body odor, because earwax and armpit sweat are produced by the same type of gland. Because this one gene has an effect on multiple traits or phenotypes, it's an example of a pleiotropic gene, because the gene affects both how wet your earwax is and how much you stink. One more thing you might find interesting, sex-linked inheritance. So we've got 23 chromosomes, 22 pairs are autosomes, or non-sex chromosomes, and one pair, the 23rd pair to be exact, is a sex chromosome. At that 23rd pair, women have 2 full-length chromosomes, or XX, and men have 1 X chromosome that they inherited from their mom, and this one little, short, puny, shriveled chromosome that we call Y, which is why men are XY. So certain genetic traits are linked to a person's sex and are passed on through the sex chromosome. Since dudes don't have 2 full chromosomes on pair 23, there may be recessive alleles on the X that they inherited from their mom that will get expressed, since there's not any information on the Y chromosome to provide the possibility for a dominant allele counteracting that specific trait. Take, for instance, balding. Women rarely go bald in their youth like some men do, because it's caused by a recessive allele located in a gene on the X chromosome. So it's rare that women get 2 recessive alleles, but men need just 1 recessive allele and don't baldy bald. And that allele is on their X chromosome, which they got from mom. But was mom bald? Probably not. And where did mom get that allele on her X chromosome? Either from her dad or her mom. So if you're bald, you can go ahead and blame it on your maternal grandmother, or your maternal great-grandfather, or your maternal great-great-grandfather, who probably went bald before he was 30. So, genetics, you guys. Resistance is futile. Thanks to my brother John for sharing his personal genetic information with us, and also his face and voice and all of that stuff. That was very nice. And think of us next time you swab out your ears. Actually, they say that you really shouldn't do that, because you have earwax for a reason, and you might poke your brain or something. Okay, that's the last time I'm mentioning earwax. Review! Click on any of these things back to that section of the video if you have any questions. Please ask them in the comments.
Okay, so that was a great overview of not only what Mendel did, but what we're going to be talking about in the next couple videos. So in that crash course video, Hank talked briefly about Gregor Mendel. So Johann Gregor Mendel, he's considered the father of genetics, and like the video talked about, he was an Austrian monk, or Hungarian Austrian monk. So he lived in this monastery, and basically he played around in the monastery's garden and manipulated these pea plants. So he actually they counted it up, and it was like thousands of pea plants that he was experimenting on. So he was very, worked with these pea plants, and he was very scientific minded, even though he was a monk. And so he, again, being this scientific minded monk, he developed what we consider now as a model system. So a model system is a species or biological system used to study a specific biological phenomenon to gain understanding that will be applied to other species. Now, he didn't consider this a model system at the time, but what he used, as pea plants, we can take that information that he gained from the pea plant and apply it to any species, essentially. So even us humans, we can apply his understanding of genetics to us. So with what he learned, he typically looked at what's called discontinuous variation. So discontinuous variation is variation in a characteristic in which individuals show two or a few traits with large differences between them. So for example, with pea plants, they come in two different flower colors. They come in violet or white, at least this particular species that he was working with. And so that's, two distinct variations. They either come out as white color or violet color. Okay, there's no in between, there's no, red color or orange color, it's just those two colors. So that's discontinuous variation. So that's a lot easier to work with than, say, continuous variation. So continuous variation is when we have variation in characteristic in which individuals show a range of traits with small differences between them, such as height. So, and we'll talk more about that type of variation later on. So in the crash course video, they talked about polygenic inheritance or genes. So poly means many, genic means genes, so many genes contributing to a particular trait. So we do have genes that can contribute to one single trait, such as our height. And because of that, you have a lot of variation within it. So, and we also have environmental things that affect our height as well. But think about things like your skin color, your eye color, your height, where we have many different variations of it. There's not just two eye colors. There's not just, two skin colors. There's not just, short or tall. There's variations of our height. And so that's considered continuous variation. But with discontinuous variation, you just have two or a few variations of it. Okay, so flower color, you have white or violet. Seed color for the pea plants, you have yellow or green. So he mainly worked with that type of variation, not with continuous variation in his model system. So it's a lot easier to figure out these inheritance patterns when you just have two distinct variations of a single trait. Okay, so we're just dealing with that type of variation in his experiments. We're not dealing with continuous variation right now. All right. So let's take a look at his crosses. So Mendel's crosses, these, by the way, these pea plants that he worked with pea plants because, for one thing, they naturally self-fertilize. Now, what that means is that they possess both male and female parts to them. So if you know anything about flowers, flowers are the sex organs of the plant. So if you think about a flower, if you ever look at a flower in detail, the pollen part is it contains the sperm of the plant. Okay, so that's the male gametes is contained within what's called the anther. So the anther has the pollen, and then at the base within the flower, I wish I had a better picture. So right here on the pea plant, you can see kind of where the pollen would be like on the anthers, and you have like the little pollen dust. That's the male part, the sperm part. And then deep inside the flower is where the ovary would be, which contains the ovule, which contains the eggs. Okay, so that would be the female part. So this plant could self-fertilize. Okay, so the sperm of this plant could fertilize the eggs of this plant. Okay, so that's one characteristic of the pea plant, which is one reason why he worked with pea plants. And so since they can self-fertilize, they could produce true breeding varieties, which means that if this self-fertilizes, it could produce offspring that look just like the parents. Okay, so if this plant self-fertilized, it would always produce violet flowers in its offspring. Okay, so it would create what are called true breeding varieties, which means that if it always self-fertilized, it would always produce violet flowers. Okay. Another reason why he used pea plants is that they're fast growing. Okay, so they you can get like several generations within a season. So they would, grow fast. They would produce flowers fast. They would pollinate and the flowers quickly, and then produce the seeds really quickly. And then you could take the seeds and then which are the little peas, and then you can plant them, and then the peas would grow really quickly and then flower really quickly. So you can get several generations really quickly within a growing season. So they're fast growing. And then finally, you can produce a lot of peas. Okay, so they produce large quantities of seeds. So they're really useful to work with. So in his experimentation, they're very, it's a very good plant to work with. Okay, so those were some reasons why he chose pea plants to work with as opposed to another type of plant. Okay, so let's take a look at the experimentation that he used. Okay, so he performed what are called monohybrid crosses. So mono means one, hybrid would be a basically crossbreeding. Okay, so he took he mated two true breeding individuals with different traits. So here's an example of a monohybrid cross. So he took one true breeding variety of a violet flower and crossed it with a white flowered true breeding variety. Okay, so this violet flower plant, if it self-fertilized, it would always produce violet flowers. Okay, it would always produce violet flowers. So he would take like the pollen of this flower and fertilize the ovules of this flower. Okay, so he would cut off the pollen of this flower so that it can't fertilize itself. So he would cross pollinate it. All right, and vice versa. All right, so this generation up here, he would label as the P generation. So think of the P generation as the parent generation. So these would hybridize. So he would hybridize the true breeding plants. And then he would, allow them to go to seed and plant the seeds and see what grew. Okay, so the offspring all came up with purple flowers. So the F1 generation, think of as the first generation from the parents. It actually stands for filial generation, but I always think of it as the first generation from the parents. They all had purple flowers. So that was something that he found kind of interesting. So even though one of the parents had white flowers, all of the offspring have purple flowers. So that was one thing that he observed. Okay. And then he allowed the F1 generation, these guys, to self-fertilize. Okay, so they were self-fertilizing. And then he took the seeds from the F1 generation and planted those. And he noticed that about 75% of the offspring of the F1 generation, which he labeled as the F2 generation, so the second generation off of the parents, were purple, but about 25% were white. So even though that these guys, their parents, none of them were white, about 25% of the offspring of the parents were white. Okay, so it's almost like the white color skipped a generation. So you notice how the grandchildren had some white in them, but the parents here, or the children of them, never had white. So we can see that the F1 generation, none of them had the white color, but the parent generation and the F2 generation showed white coloration. So he noticed that observation, okay? So the white trait or variation skipped a generation, okay, in this monohybrid cross. All right, so that's one thing that he was able to notice in his monohybrid crossings. All right. So with his garden peas, he was able to reveal some basic understanding of heredity. So in 1865, he published the results of basically 7 different characteristics, okay, of these pea plants. So he, you can look at it. These are all of them. You don't need to memorize them, by the way. But, he saw that with pea plants, they have 2 distinct variations of seed shape. So you can see that there's, round versus wrinkled, and colors, they come in yellow and green. And then we already talked about flower color. They come in purple and white. And then you can see like pod color, pod shape, whether they're tall or dwarfed, and where the flowers come out, if it's in the middle or if it's at the tips. So based on that information, he came up with some terms. So a trait is a variation in an inherited characteristic. So there's the round trait versus the wrinkled trait, okay? There is yellow versus green. There's purple versus white. There's inflated versus constricted. So these are all kind of variations of the seed shape characteristic, okay? Another thing that he noticed is that it doesn't matter if when he conducted let me just go back a slide. When he did this part, if he took the sperm of this flower and fertilized the egg of this flower, or reversed it, took the sperm of this flower and fertilized the egg of this flower, he still got the same results either way. So that's called a reciprocal cross. So a reciprocal cross is just basically where you took the respective traits of the male and the female in one cross. You could reverse it and do, the female and male in the other cross. Okay, so it doesn't matter if you're taking like the sperm from the round plant and fertilizing the eggs of the wrinkled plant, you're still going to get the same results as if you do the sperm from the wrinkled plant and fertilize the eggs of the round plant. You'll still get the same results no matter what, okay? They'll be identical. So it doesn't have anything to do with the gender. It's the same either way. So that's called a reciprocal cross, okay? So, it's not what we call sex-linked, I guess we can say, okay? It's found on the other chromosomes, all right? So based on all of that information, what we notice is that one thing we do see is that some of the traits do show up more often than others. So we do see that the round trait is more common than the wrinkled trait, okay? So that means that one of the 2 traits would disappear completely from the F1 generation and then reappear in the F2 generation in a ratio of the 3-to-1. So for example, the white trait, it disappeared in the F1 generation, but then it reappeared in the F2 generation, but only at 25%. Okay, so there's a 3-to-1 ratio. There's 3 times more violet flowers than white flowers. And so based on that information, he found that there is a dominant trait that kind of masks the expression of a recessive trait. Okay, so the dominant trait describes a trait that masks the expression of another trait when both versions are present. So in the F1 generation, both versions must be present. Okay, there must be a dominant allele or a dominant trait and a recessive trait. But the dominant trait must mask the expression of the recessive trait. So there must be a violet trait here and a white trait here in this flower, but the violet trait is masking the expression of the white trait. Okay. The recessive trait describes a trait whose expression is masked by another trait when the alleles for both traits are present. Okay. So that means that in this parent generation, let me get my pen out. What Mendel proposed is that 2 copies of the trait for each characteristic are present. Okay. So in this P generation, this violet flower has 2 copies of the violet flower trait. Okay, so I'm just going to do V for violet. Okay, so there's 2 copies of this violet color. Okay, so since this violet color is true breeding, that means that this violet plant got, its parents were violet color, its parents were violet color, its parents were violet color, so it's a true breeding variety. So both of its genes are violet. So big V, big V. Okay. This guy over here is a true breeding variety as well, but it's recessive. Okay. So it got its traits from its white flowered parents, who got its traits from its white flowered parents. So it's always white flower. So I'm going to do a lowercase v. It's hard to do a lowercase v. I'll do it like that. Okay. So then remember meiosis, we take a diploid cell and we make haploid gametes from it, right? So we're only passing on one of these genes to the next generation, or one of these V's to the next generation. So I'm going to put, so 1 copy goes into the sperm, okay? 1 copy goes into the egg, right? Or vice versa. So this V is going to go down here into this plant. Okay, this V will go down here into this plant. Right? So this flower right here has a big V from this parent and a little v from this parent. Okay, and because of that, it's got a dominant allele and a recessive allele. But we know that the recessive allele is masked by the dominant. Okay, so that's why it's showing purple flowers. Okay, and then if it self-fertilizes, okay, sometimes, we see in the video that we saw that a Punnett square was set up. So I can just go ahead and show you what a Punnett square looks like, okay, from the video. We'll have lots of practice doing Punnett squares later on. You're not expected to really know how to set one up just yet, but since we're here, we'll do one. With a Punnett square, what we do is we take these genes here. So since this is self-fertilizing, okay, this is both parents, right? Okay, so this could be, the sperm undergoing meiosis. So the sperm undergoing meiosis, we have a big V going here and the little v going here. The eggs here are also or the female part is undergoing meiosis as well to make the eggs. Okay, so the big V goes up here, the little v goes up here. All right, and then to set up the Punnett square, so basically mom's alleles go up here, dad's alleles go down here, or vice versa. You could put mom's alleles here, dad's alleles here, or traits here. And then basically you just bring the V's across and the V's down. So if I bring this V across, big V, this V goes across, little v, bring this V down, big V, this V down, little v. These are the results, okay? So I have big V, big V, little v, big V, little v, little v. So this guy right here, notice, is little v. This one does not have a dominant allele, so this one's going to be white, okay? These all have dominant alleles, so all of these will be purple. So that's why we have a 3-to-1 ratio. Okay, there's a 3 or 75% chance that we're going to get purple flowers, 25% chance that we're going to get white flowers. All right, so that's where we get that 3-to-1 ratio. And you know, we'll play around with Punnett squares a lot in the next video. Okay, so for now, you don't really need to know how to set up a Punnett square, but just realize this is where we're getting that 3-to-1 ratio. Okay, all right. So let's do a review question to make sure we're understanding all of this Mendel's experimentation. So imagine that you are performing a cross involving seed color and garden pea plants. What trait would you expect to observe in the F1 offspring if you cross true breeding parents with green seeds and yellow seeds? Okay, and remember, yellow seed color is dominant over green. So yellow seed is dominant over green. So we are crossing yellow seed to green seed of true breeding varieties. What do we expect to see in the F1 generation? Is it A, only yellow-green seeds, B, only yellow seeds, C, a 1-to-1 ratio of yellow to green seeds, or D, a 1-to-3 ratio of green to yellow seeds? If you said B, I would agree with you. We would only see yellow seeds. Okay, so if these are true breeding varieties, that means that this yellow seed, if we cross this yellow seed that only makes yellow seeds if it self-fertilizes, and we cross that with this green seed that only makes green seeds if it self-fertilizes, these are the parent generation, right? And if we cross them, the F1 generation would only produce yellow seeds, because at that point they would have a dominant allele and a recessive allele, okay? And so the dominant would mask the expression of the recessive, and so they would only have yellow seeds in that F1 generation, just like this scenario, okay? So instead of violet and white, imagine you'd have yellow seeds and green seeds, so they would all produce yellow seed F1 generation. Okay, so B is the correct answer. So the only thing we're changing here is that we're using this example instead of this example. Okay, here's another question. Imagine that you are performing a cross involving seed texture in garden pea plants. You cross true breeding round and wrinkled parents to obtain F1 offspring. Which of the following experimental results in terms of number of plants are closest to what you expect in the F2 progeny? Okay, so you are performing a cross involving seed texture in garden pea plants. So you cross true breeding round and wrinkled parents to obtain the F1 offspring. Okay. By the way, if we go back, round versus wrinkled, round is dominant to wrinkled. Okay, just to give you an idea. So we have our round and wrinkled parents. Now we have the offspring. Now, what do you expect in the F2 progeny? Do we expect all round seeds, about 810 of them? Do we expect all wrinkled seed, 810? Do we expect about even numbers of round versus wrinkled seeds? Or do we expect about 610 round seeds to 190 wrinkled seeds? If you said D, I would agree with you. Okay, 610 to 190, so that's, about 6 to, 2. That's about, almost a 3-to-1 ratio, right? Close enough. So in the F2 progeny, this is about a 3-to-1 ratio, as closer to 3-to-1 as any of these. Okay, so this is the dominant to the recessive. So this is the correct answer in our F2 generation. All right. Okay, so we are done with our first section of our Patterns of Inheritance, Chapter 7. So by now in Mendel's experiments, you should be able to explain the scientific reasons for the success of Mendel's experimental work, as well as describe the expected outcomes of monohybrid crosses involving dominant and recessive alleles. Okay, so in our next video, we're going to get into the laws of inheritance in 7.2.